Good afternoon, and welcome to the annual Memorial Sloan Kettering Convocation and the commencement of the Gerstner Graduate School of Biomedical Sciences. I'm Craig Thompson, President and CEO of MSK. We want to thank everyone for taking the time to join us for today's celebration. This is MSK's 41st convocation and the 9th commencement of the Gerstner Sloan Kettering Graduate School. But this is the first time we have done this ceremony remotely. So we'll be doing things a little bit differently today. I'm joined by a few of my colleagues today in Zuckerman Research Center auditorium, where we are spread around the room in keeping with social distancing. I am joined by Joan Massaguet, our provost and the director of the Sloan Kettering Institute. Lisa DeAngelis, our physician-in-chief and chief medical officer, and Michael Overholzer, Dean of the Gerstner Sloan Kettering Graduate School. In this virtual event, we are pleased to recognize the graduates of our PhD programs and our annual award recipients. We are particularly excited to have the 8 GSK students who will receive their PhDs in cancer science today. I also want to give my congratulations to the 34 Weill Cornell Sloan Kettering graduate students who have earned their PhD degrees while working in MSK laboratories this year. We're extremely proud of your accomplishments. In addition to the graduates and award recipients we will be recognizing today, I also want to acknowledge other award recipients from this past year. Maria Jason, member of SKI's Developmental Biology program, was named the winner of the Shaw Prize in Life Sciences and Medicine. She was honored for her work showing that localized double-strand breaks in DNA stimulate recombination in mammalian cells. This work helped lead to the development of tools which now edit specific sites in mammalian genomes. This past month, both Chris Lima, chair of SKI's Structural Biology program, and Scott Keeney, a member of SKI's Molecular Biology program, were elected to the National Academy of Sciences, joining 13 other MSK faculty as elected members. Also in this past year, Scott Lowe, chair of SKI's Cancer Biology and Genetics program, was elected to the National Academy of Medicine for his pioneering work on tumor suppressor gene networks. He is one of 25 MSK faculty who are members of this prestigious organization. Michelle Sadelain, director of the Center for Cell Engineering and a member of SKI's Immunology program, received the Jacob and Louise Gabay Prize in Biotechnology and Medicine for his groundbreaking research on CAR T-cell therapies. Charles Sawyers, chair of the Human Oncology and Pathogenesis program, was awarded the inaugural STAT Biomedical Innovation Award, recognizing a top researcher whose work has helped define their field and helped patients. Ann Zauber, a member of the Department of Epidemiology and Biostatistics, was recognized with a Life Achievement Award for her work on colorectal cancer screening. And Adrian Bois, Yael David, and Alex Kensis each received Pershing Square Sohn Prize for Young Investigators, awarded to exceptional young scientists who have made innovative ideas part of cancer research. It's been an incredible year at MSK. I'm particularly pleased to also report that the crucial work that was paused during our COVID-19 response is now resuming in phases across our research enterprise. To begin the presentations of this year's Research and Clinical Awards, I would like to turn the podium over to Joan Massaguet, Director of SKI. Joan.
Thank you, Craig. And thank you to everybody for turning in on this special event. Although we are unable, because of the circumstances, to have our traditional gathering to celebrate our graduates and awardees, we are very, very pleased that you can join us in this way, in this virtual way, to celebrate them nonetheless. We have a few new awards this year. I'd like to thank the generous supporters who help in the creation of these awards through their generous funding. And now it is my pleasure to introduce this year's Research Award winners. Detailed information on each of these awards and awardees can be found in the 2020 Convocation and Commencement webpage, where you are viewing this event right now. The Doris Hutchinson Fellowship recognizes outstanding research done by a graduate student working under the mentorship of an MSK faculty member. This year, the fellowship is awarded to Laura Menokal. Laura is a PhD candidate in the Weill Cornell Graduate School of Biomedical Sciences. She's conducting her research in the lab of Andrea Schettinger. Her research is focused on how memory T cells differentiate in the context of tumors in cancer. The Barbara and Stephen Friedman Fellowship Award was established with an endowment from the MSK board member Steve Friedman through his Friedman Family Foundation. It supports predoctoral students. The award is being given to Disha Deep. Diksha is an MD PhD student in the tri-institutional Rockefeller, Cornell, Weill Cornell, Sloan Kettering program, tri-institutional program. She's doing her thesis in the lab of Alexander Rudensky, and she's studying how dendritic cells respond to fungal infections. Next is the Bruce Charles Forbes Fellowship Award, established with a gift from MSK board member Steve Forbes, Jr. It also honors a predoctoral student. This year, the award goes to Zhongmin Wang. Zhongmin is a Gerstner Sloan Kettering student who is conducting his thesis research with Dr. Rudensky in his lab as well. He studies how regulatory T cells function under inflammatory conditions. The General Atlantic Graduate Research Fellowship Award provides support for a promising researcher to pursue his or her education and key research interests that integrate scientific priorities with important clinical needs. This year, General Atlantic Fellowship is awarded to Inbal Caspi. Inbal is a Weill Cornell graduate student in the lab of Danwei Huangfu. Her research is on glucose transporters in human pancreatic cells. The postdoctoral research awards recognize outstanding accomplishments and promise by research fellows or research associates who have been at MSK for at least 1 year and have published a significant research paper as first authors. This year's postdoctoral research award recipients are Sumru Bain and Marcus Rossetti. Dr. Bain is studying brain regeneration in Alexandra Joyner's lab in the developmental biology program. Dr. Rossetti is studying targeted therapies for cancer in the lab of Scott Lowe in the cancer biology and genetics program. We are pleased today to recognize the first fellowships for the new Marie-Josée Kravis Women in Science Endeavor, alias WISE, at MSK. This initiative is supported by longtime board member Marie-Josée Kravis, and the WISE Fellowships have been granted to 3 promising trainees. The first WISE Fellowship award goes to Sahana Rao, a graduate student in the tri-institutional PhD program in chemical biology. She's doing her thesis in the lab of Daniel Bachovchin in the chemical biology program. Her research is focused on the role of inflammasomes in the anti-cancer immune response. Next is Lind Miles, a postdoctoral fellow in Ross Levine's lab. Lind is investigating the role of clonal architecture and evolution in acute myelogenous leukemia. Finally, there is Mohita Malai Tagore, a postdoctoral fellow in Richard White's lab. She's studying the role of melanoma microenvironment in tumor progression and metastasis. The Tri-Institutional Breakout Prize for Junior Investigators was established by leaders from the Rockefeller University, Weill Cornell Medical College, and Memorial Sloan Kettering Cancer Center. It recognizes exceptional current postdoctoral trainees at each of the 3 institutions who have made significant research contributions. The winner from Memorial Sloan Kettering is Manu Sethi. Dr. Sethi is studying single-cell analysis and computational biology in the lab of Dana Peer in the Computation and Systems Biology program. Every year, the Louise and Alston Boyer Young Investigator Award for Basic Research honors someone at the associate level or below who has demonstrated great promise and accomplishment in laboratory investigation. This year's recipient is Andrea Schettinger, already mentioned. Dr. Schettinger joined SKI in 2015 as an assistant member in the immunology program, following her postdoc at the University of Washington. Since coming to SKI, she has made important contributions to cancer immunology and immunobiology research, especially studying the role of T cells in cancer and their regulation and disruption of their functions in the context of tumors. She's won many other awards, including the NIH Director's New Innovator Award in 2017. The Catherine Birkin Judd Award recognizes an individual who has made great advancements in the understanding of cancer through basic discovery. This year is awarded to Ronald Vail. Dr. Vail is Executive Director of the Howard Hughes Medical Institute's Janelia Research Campus and is an HHMI Vice President. His research is focused on molecular motor proteins and the role of these proteins within cells. He's also involved in many activities that benefit the wider scientific community in the dissemination of science. Dr. Vail will give his award lecture on Monday, June 1st, as part of MSK's Virtual Science Spotlight series. And now, I'd like to introduce Dr. Lisa De Angelis, who will present this year's clinical awards. Lisa?
Thank you, Joanne, and good afternoon and welcome to this wonderful celebration. I'm thrilled to be able to announce this year's clinical award winners. We are so proud of all your accomplishments. The Louise and Alston Boyer Young Investigator Award for Clinical Research honors physicians who have demonstrated great promise and accomplishment in clinical investigations. This year's award winner is Alex Kensis, an associate member of SKI. He is a pediatric oncologist who treats children and young adults with leukemias and refractory solid tumors. His research has led to new therapeutic targets in cancer cells and understanding of the mechanisms by which cancer cells can evade new treatments and improve strategies to overcome treatment resistance. I'm pleased to present him with the Boyer Award. The C. Chester Stock Award lectureship is given annually to an individual who has made significant contributions to the advancement of cancer therapy. This year's recipient is Catherine A. High, MD. Dr. High is a professor emerita at the Perelman School of Medicine at the University of Pennsylvania, an attending physician with the Division of Hematology at Children's Hospital of Philadelphia. Dr. High, an accomplished physician scientist, presented her lecture, Turning Genes into Medicines: What Can We Learn from the Development of Gene Therapy for Genetic Disease, last week as part of our Science Spotlight series. You can see a replay of her May 20th lecture on mskcc.org. I'm proud to present Dr. High with the C. Chester Stock Award. The Willett F. Whitmore Award for Clinical Excellence is presented to a clinician whose talent, dedication, and compassion reflect the standards set by Willett F. Whitmore during his more than 46 years of service at Memorial Sloan Kettering. We're pleased to present this year's Whitmore Award to medical oncologist Carol Agajanian. Chief of the MSK Gynecologic Medical Oncology Service. Dr. Agajanian is renowned among her patients for her compassion and deep commitment to their care. She is also a pioneer in the treatment of rare cancers, such as treating advanced cases of gestational trophoblastic disease, a rare tumor that develops from fetal tissue after a pregnancy. She is also a widely recognized superb teacher and mentor who's committed to developing the next generation of oncologists. Today, we recognize her dedication to her patients and trainees and her clinical research accomplishments. We are all privileged to have her here at MSK. And I'm pleased to present Dr. Agajanian with the Willett F. Whitmore Award for Clinical Excellence. The Society of Memorial Sloan Kettering Prize recognizes individuals who have made significant or groundbreaking contributions to pediatric oncology. Further, it highlights the long-standing and future-facing partnerships between the Society and MSK and affirms the global leadership of MSK in the field of pediatric oncology. This year, we are very pleased to have two Society Prize winners, Carl H. June, Director of the Center for Cellular Immunotherapies at the University of Pennsylvania, and Michelle Sadelain, Director for our Center for Cell Engineering here at MSK. This award recognizes their pioneering contributions to CAR T-cell therapies, a life-saving advance in treating childhood leukemia and other diseases. Their decades of commitment to developing adaptive cell therapy give many children and their families new hope. I am proud to present the Society of Memorial Sloan Kettering Prize to Drs. Carl June and Michelle Sadelain. Congratulations to all of this year's winners. Thank you for your many contributions, and we're proud to celebrate your achievements today. I will now turn it back to Craig Thompson, who will introduce our honorary degree recipient and this year's commencement speaker, Dr. James Allison.
Well, thank you, Joanne and Lisa. I'd like to echo your congratulations to all of this year's award recipients. As Lisa said, it's now time to introduce this year's honorary degree recipient and commencement address speaker, Dr. James Allison. Jim. In truth, Jim probably needs no introduction. His work has transformed the way we think about the immune system as an ally in the fight against cancer. The results of his pioneering research are saving lives right now at MSK and at hospitals throughout the world. In 2018, Jim received the Nobel Prize in Physiology and Medicine, along with Tasuku Honjo, for their work on immune checkpoint blockade. The Nobel Assembly called the work an entirely new principle for cancer therapy. The prize recognized Jim's basic science discoveries concerning the biology of T cells, the soldiers of the adaptive immune system. In particular, Jim showed that by blocking the brakes that turn off activated T cells, he could free T cells to persistently attack cancer cells. And they can persistently attack, as he showed in a pioneering paper in 1996, that they would persistently attack cancer cells until they were completely eliminated from the body. This finding transformed the field of immuno-oncology. We are proud that Jim led the translational efforts to introduce his discoveries into the clinic here at MSK when he served as chair of immunology at SKI from 2004 to 2012. Working alongside him was our own Jed Yelchik, now chief of the immuno-oncology service. Jim is currently the chair of immunology and the executive director of immunotherapy platform at the University of Texas MD Anderson Cancer Center. He is also a scientific director at the Parker Institute for Cancer Immunotherapy. Among Jim's many recognitions, he is a member of both the National Academies of Sciences and Medicine. He has received the American Association of Immunologists Lifetime Achievement Award, the Lloyd J. Old Award, the Lasker DeBakey Clinical Medical Research Award, and many other honors. Today, we are proud to award James P. Allison the honorary degree of Doctor of Science at the Louis V. Gerstner Jr. Graduate School of Biomedical Sciences at Memorial Sloan Kettering Cancer Center. Jim, this is the diploma. It will be with you shortly. Jim is a role model for the graduates we celebrate today. His career is a testament to determination, the value of collaboration, and the foundational importance of basic research. It is with deep appreciation that I present Jim Allison, who joins us from deep in the heart of Texas. Jim, I turn it over to you for the commencement address.
Thank you, Craig, and thanks for that honorary degree. I don't suppose it comes with a stethoscope, but anyway, I'm very honored to get that, particularly from Sloan Kettering and from you, Craig. And I'm also glad today to have the honor to offer my congratulations on the graduate students on their finishing, or maybe I should say surviving, the rigorous program and preparing themselves for careers as scientists. I realize this isn't probably the way they wanted to do this. You know, after all these years, they probably were looking forward to having their families there and their friends and walking across that stage as their name was called. But, you know, to mark the beginning of this new chapter in your journey in life. But unfortunately, COVID-19 prevented that. Despite that, I trust that this will be a fine day for all of you anyway. I know the GSK program well. As Craig mentioned, I was on the faculty there when it was founded in 2004, participated in the program to my departure in 2012. It's very gratifying to be back there, even though virtually in the Zuckerman Auditorium, which actually I've never been in physically, to see how successful this program's become. I looked over the research projects of the students that are finishing and found that they really represent work at the very cutting edge of basic and translational cancer research, from CRISPR mutagenesis of xenografts to studying mutations of proteins that affect epigenetic control of cancers and all the other ones that were there. Quite an impressive array. So it appears to me that the GSK is indeed successful in its goal of guiding the development of scientists with the potential to be tomorrow's leaders who contribute to minimize and eliminate at least some of these diseases which scourge mankind. In addition to congratulating you, I'd like to take a few minutes to make some observations, offer some advice, and tell you some stories that might be useful, some of which might also help the non-scientists, if you have any, that are sitting with the other graduates among your friends and relatives that understand what we do. You've reached a major milestone, and what is probably the first major milestone, or maybe the second if you count your undergraduate degree, in your journey so far. I think that for many of you, graduate school may have been the best time in your life so far. Although when I said that once, one of my graduate students said, "Really? Is this as good as it gets? I think I'll shoot myself." But anyway, it was great for me. And why do I think it would be great for any aspiring scientist? And I think that's because we don't choose science so much as science chooses us. I think most of us start down this pathway because we're curious and we want to know how things work. In our case, living things. And graduate school is really the first time we have a chance to do that. When you were a kid, you may have had a chemistry set in your garage or a microscope in your bedroom that you spent time with. And in high school, you might have liked math and science courses more than the rest. And by the end of high school, you may have had a strong interest in science, but you know, there were choices to be made for many of you. And one of them, I think, was a choice that many of us faced, because my dad was a small town doctor who made house calls. In fact, he was probably the first immunologist I met, because whenever a kid in the neighborhood, any of the families he cared for, developed one of the then very frequent childhood diseases that still occurred frequently because there weren't any vaccines then, he would have me sit on the sofa with them during his examination in the hope that I'd get whatever it was that they had while young and be spared the much worse sickness that could come with the illness in adults. Also, if it was during the summer vacation, he hoped, of course, that I would get it then and not miss any school. But he really wanted me to consider medicine. In fact, I entered the University of Texas as pre-med. As I began to work in a laboratory, and after my experience in a summer course in science and in biology at the University of Texas, I realized that there's a fundamental difference between practicing medicine and being a scientist. Watching my father all those years, I realized that a doctor needs to have a head full of facts, stuff their head full of facts so that they can draw upon them to generate a treatment algorithm according to how a patient presents. And it better be correct, or the worst you could do is not hurt the patient. But I certainly didn't have enough discipline for all that cramming and memorization. As I progressed at UT, working in a lab doing an undergraduate research program, I began to realize that scientists have a very different job than medical doctors. Scientists should generate new ideas and test them. Many, if not most, will be wrong, especially if that work is on the leading edge of a field. Thus, we're really supposed to be wrong a lot, unlike a physician. It's sufficient only to be correct some of the time, hopefully about important things, in order to keep the field moving and advance knowledge. In college, as an undergraduate, you took math, you took physics, maybe gravitated towards biology. And maybe you began to seriously think of becoming a scientist, making your living doing this. And that's very cool, but probably a little scary. But you guys obviously decided to take that next step anyway. And it's in graduate school where you really begin to learn how science works. It's a heady atmosphere. You can immerse yourself surrounded, probably for the first time in your lives, with your colleagues, very smart, like you are. And though their particular interests may differ from yours, nonetheless, they share and exult in the idea of knowledge and science. And sometimes when things aren't going well, and there are such times, but hopefully not that many, these are people with whom you can commiserate. There are also more senior graduate students, postdoctoral fellows to help guide you. And of course, there's the faculty. They teach the necessary classes and with the help of postdocs, of course, and run lab meetings and help guide you and give you access to their special experience and knowledge, when they're around anyway. But the lab, all the equipment and the instruments and the doodads and thingies that are really fascinating. And all right, you've got them all, and you can do what you want. You've got a stipend, and you can begin the serious and difficult task of actually becoming. And that's what you guys have just been through. And as you gain more experience in your specific area of interest, you begin to see whether areas of insufficient or shallow knowledge and begin to ask questions that remain, important questions, hopefully. You learn to generate a hypothesis, design an experiment to test it, and do the experiment to get the answer. Easy, right? I mean, that's the definition of science. Unfortunately, you probably quickly learn that that's not actually so easy, and that's not really the way it works. Too often, the experiment doesn't give a clear answer, or worse, it generates two new hypotheses. Science is hard, very hard, because you mostly are learning what is not correct. My graduate days and undergraduate days, actually, I was a biker, not a bicycle, but in the sense that my main means of transportation was a British twin, a 1974 Norton 850 Commando. During this time in school and riding this motorcycle, I read this interesting book called Zen and the Art of Motorcycle Maintenance, believe it or not, by a guy named Robert Pirsig, who was a PhD biochemist, ex-scientist who turned a philosopher biker. Anyways, this thesis was that, when you're working on a motorcycle, you're really working on yourself. But that wasn't the interesting thing about it. The interesting thing was he had this thesis that since the answer to the test of each hypothesis led to two more, in his experience, hence science. And then each of those led to two more, hence science, rather than providing answers, was actually leading to ignorance and leading to that at a geometric rate. Well, while it's no doubt a good thing that a person quit science, the situation is not really that bleak. If an experiment's well designed, properly controlled, and performed carefully, there'll usually be at least part of an answer. Maybe no, and you were wrong. Or it may be that you were not completely right, completely clear, but it could help you design additional experiments. But in any event, there are three things to remember that I've learned about data and science. The first is that data are like your children. You may not like them very much, but once you've got them, even if they're negative all the time. But you must take them into account as you proceed. Another facet of data is that it should also be like your significant other, your lover in a way. You need to spend lots of time with it, trying to understand your data. Listen to the things the data are saying, like jewels. Look at them at different angles and try to take different lights from them and learn the secrets. Very often, more than if they're right or wrong, you can see something in them. Maybe in your controls, maybe in one of the test groups, you can see something that leads you down another path if you just pay attention. And you need to do that because nature doesn't yield her secrets easily. And the third one is the data in the experiment should have been designed to answer the question at hand, and very often they don't. For example, in my own background, and Craig was involved in this too, we knew that CD28 was a receptor on T cells that was required for a co-stimulatory signal. It was needed in addition to the antigen receptor signal for full activation of T cells. But it was known at that time that there was another gene, CTLA4, that was highly homologous to CD28. The only thing that was known about at the time was that it was not expressed in naive T cells, rapidly induced upon T cell activation, and bound the same ligands, B7, the same counter receptors as CD28. So the question, of course, was what was the function of this molecule? And of course, this was a subject of a lot of work by a significant cadre of immunologists, including myself, Craig, Jeff Bluestone, and others, Peter Lindsley, and others. And just as my lab finished generating a monoclonal antibody to the mouse homolog, we're beginning our functional studies, a paper appeared purporting to show that CTLA4 was another co-stimulatory like CD28. And it had a higher avidity, and its job was to sustain co-stimulation as the level of the ligands was diminishing on dendritic cells and keep T cell going. And it did this by showing that if you add antibodies to T cells undergoing activation by T cell and TCR signaling, it increased proliferation, cytokine signaling, et cetera, et cetera. So Max Krummel, who was the graduate student in my lab, his thesis project was, what is the function of CTLA4? He and I were initially disappointed. We felt that we'd been scooped. Indeed, the data in the paper were absolutely consistent with a positive role for CTLA4. However, as we look closely at the data and more carefully at the experiments, we realized that the data actually were equally consistent with the opposite possibility. And that is that CTLA4 was an inhibitory molecule, and that the increase when you added the antibody was because it was blocking a negative signal. And their experiments couldn't tell the difference. And so it led me to question why they were able to conclude the title of that paper as strongly as they did. Anyway, we looked at it. We designed two independent experiments that could give unequivocal answers to show, one or the other, and first showed that it was indeed had a co-inhibitory function, if you want to use that word. In other words, it was a negative molecule, the first one that was identified on T cells. This then, of course, led to the development of new strategies for manipulating T cell activation. It was the origin of the immune checkpoint blockade approach to treating cancer that Craig mentioned. So the point I'm making here is that a killer experiment is worth dozens of consistent results, and that's what you need to work for. Anyway, most of you, I expect, are going on to postdocs. This next phase in your development should be even better than graduate school. You have the skills, you have the basic knowledge, perhaps access to more advanced techniques and better collaborators, more advanced collaborators. You'll learn there to develop your own ideas with progressively decreasing guidance from your mentor and others, and dive more deeply into the areas that interest you. At this time, you'll be free from the distractions that'll burden you for the rest of your career, whatever you do, such as the necessity to get funding, administrative duties, teaching, workplace politics, all the noise that comes with any job, whether it's in academia or pharma. So I would advise you to do absolutely all you can to, find your way towards your future during your postdoc. And I want to point out too that I think there's a dirty little secret about what keeps scientists working when the going is tough. And we know there's no instant gratification in doing science. The little secret is that ego boost, the high you get for being the first, the very first, and for a while, the only person on the planet who knows the answer to an important problem. It's as a postdoc that you will get to experience this. Later on in your career, you'll best be second or maybe more after your graduate student or your postdocs learn this. The next period should be a time for you to fly, extend your wings as far as you can and find something you love, are good at, and want to pursue for your career. How should you choose what to do for your career? The hottest topic in nature, science, or nature? No, an area that's being recruited heavily, not necessarily. I would suggest that passion is important. Do something that keeps you interested. I fell in love with T cells after an undergraduate course that I had, just after they were discovered, at a time when nothing was known about them. We know a lot more now, still working on them, and I'm still captivated as I'm ever. Another question is, should you be a basic scientist looking for first principles or more translationally oriented, making advances in treating cancer? Both are important and they're not mutually exclusive. Again, let your passion drive you. And I will tell you that these are the opportunities that you have as you go forward. It's going to be challenging. As you know, the COVID pandemic is going to have a big impact on the economy. I spent a lot of time this past year in Washington lobbying for the NIH budget, and I think that we did well in getting a $2 to $3 billion increase. At least that's the way it was passed. And since the NIH and the NCI are the main drivers of basic science, I think that that's a good sign. But this may be different in the future. But those of us who have been around for a while know that they're hard times, good times, and they're cyclical, and they come and go. But if you endure them, you will get through. It's, of course, made worse by we have an administration that is openly unfriendly, even hostile to science, and assaults even the existence of truth. But again, I think that we could overcome that in the future. Part of the problems with you have to be advocates for change. You have to help us create an environment, an atmosphere in which we can accomplish the things that we want. But I will tell you, even given all these challenges, the opportunities that you have are incredible. The state of knowledge now is obviously such that it's never been. The instruments that are available, the techniques that are available allow you to ask questions that we could only dream of. And you're only going to be limited by your own energy, your imagination, your passion, and your perseverance. But I think your possibilities are endless, and I have a lot of envy for you guys because the best is yet to come for you. And so I'll close there and look forward to chatting with you. I can. Thank you.
Thank you very much, Jim. I'd like to offer my deepest gratitude to our honorary degree recipient and commencement speaker, Dr. James P. Allison. My name is Mike Overholtzer. I'm the Dean of the Gerstner Sloan Kettering Graduate School at Memorial Sloan Kettering Cancer Center. I would also like to offer my congratulations today to each of our 42 graduating students who are recognized for having earned their PhD degrees. To our graduating class, today is all about you. You know, it wasn't that long ago that each of you came here to join our community seeking to learn a little bit about biomedical research. And while we tried to teach you all of the knowledge that we had at the time in your first year classes, what we really wanted to teach you about was the process of discovery, to show you how to discover new knowledge and to immerse you in this process within our community here that is so passionate about it. We also wanted to show you how the hope that some of our new discoveries will make a lasting impact on human health drives our research here at Memorial Sloan Kettering. As your time in our community progressed, each of you transitioned from learning about this process to becoming major drivers of it. You each made your own new discoveries. And you wound up teaching us through your research and through your dedication and your passion for this process everything that this next generation of great leaders has to offer. We are enormously proud of your accomplishments. I'd like to first recognize and congratulate the 34 graduates of the Weill Cornell Graduate School of Medical Sciences who performed their thesis work in Memorial Sloan Kettering laboratories. Our Weill Cornell graduates will have a separate commencement ceremony tomorrow with the Weill Cornell Medical College. But today, I'd next like to congratulate the 8 graduates from the Gerstner Sloan Kettering Graduate School who are receiving their PhDs. These are Ivan Cohen, Christopher Halton, Mark Yelchik, James Keith Jr., Nicholas Kuhn, Hannah Wise, Brittany Woods, and Yu-Chen Shi. Congratulations. We've put together a brief video so that you can learn a bit more about each of them from those who know them best, their thesis mentors.
Hi, Ivan. It's Ron Blasberg. You're a mentor at GSK for your graduate program and the PhD degree. Your program involving immune cells involved in fighting triple-negative breast cancer was really an outstanding piece of work. Now, one bit of compliment here that I think is a little bit unusual for graduate students, and that is your ability to think outside of the box. This was really a major asset that you demonstrated during your time in the lab and contributed substantially to the work progressing in the lab. We all are appreciative of this and of your contributions over the several years that you were with us. Ivan, again, best of luck. We are all appreciative of what you've done, and we know that you will have an outstanding career in science. Bye now, until the next time we meet at an international meeting.
Hi, I'm Charlie Rudin, and it's a real pleasure today to introduce Chris Halton. He's been a fantastic graduate student. Chris came to us after having substantial research experience in the past, and even as a rotating student, it was really remarkable. We gave him a cloning project that we thought would really take him the whole rotation, and he came back about 2 weeks later saying, you know, I did a one-step cloning method where I combined all these things, and he got it all done, and we sort of were like, wow, what are we going to do for the next 6 weeks? He really came with a lot of skills, he published a fantastic thesis on direct genome editing of patient-derived xenografts using CRISPR-Cas9 in a way to enable in vivo functional genetics. And this was a real tour de force in terms of sort of technical undertaking. And Chris has gone on to become a research scientist at C4 Therapeutics up in the Boston region in Watertown, Mass. And I'm sure he will continue to do great work there. So it was a terrific pleasure to have Chris in the lab. He was really a spectacular student and did beautiful work that I think has high impact for the future. And I wish him all the best and great congratulations on his graduation today.
Hello, so I'm Philip Niethammer. I'm a PI in the cell biology department of Memorial Sloan Kettering. So please let me say a few words about Mark Yelchik, which I was fortunate to have in the lab for the past 5 years or so. Following up on work of a former postdoc, Mark became interested in the question how extracellular ATP, a small nucleotide widely recognized as important wound healing and inflammation signal, mediates rapid wound healing in live zebrafish. Well, at least this was his initial motivation, because driven by Mark's strong interest in biochemistry, proteomics, and chemical biology, the project soon unfolded its very own dynamics. And Mark, just as any good scientist, went with it. Mark single-handedly established a collaboration with Ming-Bin Liu's lab to develop a novel type of ATP affinity probe to screen with quantitative proteomics for unknown ATP interacting proteins on the plasma membrane that could potentially act as novel types of ATP receptors. This project charted entirely new territory for my lab. So suffice to say that Mark's enthusiasm, experimental craftsmanship, and serendipity have been a cornerstone of the lab for the past 5 years, and it was a real pleasure and rewarding experience to be his mentor. So many congrats, Mark, and all the best for the future.
I'm pleased to introduce James Keith, who was my first and only GSK student to join my laboratory. And he performed a wonderful set of experiments characterizing the role of the microbiome in Clostridium difficile infection. And over the course of several years, he demonstrated that antibiotic treatment of mice left them with bacterial populations that impacted the immune system of the mice. And he posed the hypothesis that this might influence the severity of colitis in animals that are exposed to Clostridium difficile, and was able to demonstrate that the residual bacteria in the gut can have a significant impact on the immune system. But surprisingly, what he found was that this had a little impact on the severity of colitis, which seemed to have much more to do with the relative virulence of different strains of Clostridium than it had on the residual bacteria. I'd like to congratulate James for completing this body of work and obtaining his PhD from the Gerstner School.
It gives me great pleasure to introduce and to congratulate Nicholas Kuhn, who was a graduate student in my lab for a little over 4 years. I'm wearing my mask, as you can see, to remind Nick that he needs to be supporting the Buffalo Bills and none of those other teams that he's supported in the past. And I'll take that off right now and have that finish as a reminder. Nick worked on CD40 ligand and CAR T cells with the idea that CD40 ligand being expressed on the surface of the cell can help stimulate the endogenous immune response. Nick was wonderful to mentor, and I can honestly say that by the end of the day, I've learned as much about mentoring and as much about CD40 ligand from him than he ever learned from me. And hopefully some of that will continue as he starts his own lab, which I invariably expect to happen in the very foreseeable future. So Nick, Congratulations. You set the bar extremely high for people that follow you as graduate students and even as postdocs. And you will be missed. And it was a pleasure and an honor mentoring you through this process. Congratulations and best of luck to you in the future.
So I'd like just to congratulate Dr. Hannah Wise on her graduation this past year. Hannah joined the lab a number of years ago, and her main project focused on a set of mutations that are very common across many different cancer types, in particular in patients with bladder cancer, in genes that regulate chromatin structure and accessibility, and gene transcription in particular. So she's now a lead clinical data analyst at Flatiron Health here in New York City, where she's working with data scientists and clinicians, hoping to use some of her expertise in cancer biology and genomics and clinical oncology to understand better what mutations are doing in the clinic in a larger scale. So I'm excited that she was able to find this opportunity to really put to use many of the things she learned here as a graduate student. Really excited to see that she's enjoying that job and has, I think, found a good home.
Hi, Hannah. We're all here from the Cell Lab wanting to wish you congratulations on your graduation. Congratulations, Hannah!
Thank you, Hannah!
Great job.
My name is Ross Levine, and I have had the distinct pleasure of being the mentor to Brittany Woods, who's graduating today. There's a lot I can say about Brittany and what she's meant to the lab, to the field, and to me personally. But I'll start by saying that she's just an exceptional scientist whose contributions to the field of myeloid neoplasia, and specifically myeloproliferative neoplasms, has been tremendous. She's elucidated the role of mutant JAK2 in this important disease, including in platelets, and her insights are leading to new ideas that have both mechanistic and therapeutic implications, including, we think, in clinical trials. But more important than that, I think what has been amazing to see is Brittany's growth as a person and as a colleague. She's a spectacular scientist, an amazing person who's supportive to everyone around her, who shows tremendous strength of character and resolve, and her perseverance to really ask hard questions and solve them in the lab was not only amazing for me to see, but for everyone to see, and a model for how scientists should really do their work. So from everyone in the Levine lab, we say congratulations. We're so excited for you today, but most importantly, we're excited that you're part of our family and will always be. Thanks so much. Congratulations, Brittany. We're so proud of you.
I'm Emily Chen, member of Human Oncology and Pathogenesis Program. I am proud to introduce my trainee, Dr. Yelchik PhD, as a member of the graduating class of 2020. She joined my lab in 2015 when my lab started a new research direction on epigenetic tumor suppressors that are commonly mutated in kidney cancer. With her passion in epigenetics, Yelchik took a challenging project to investigate the tumor suppressor function of CD4, a histone methyltransferase. Yelchik was very brave to explore this new research frontier and has pioneered many experiments that have not been routinely performed in my lab. Yelchik proved herself to be a brilliant scholar. She received the prestigious F99K00 award from NCI in 2018 and will go on to complete her postdoctoral training with Dr. Neville Sanjana at New York Genome Center. I'm fortunate to have had such an excellent student in my lab, and I'm excited to continue to watch her scientific career blossom. Congratulations, Yelchik.
And congratulations to all of the graduating class. And to all of you in the audience, it is now my pleasure to present the GSK graduating class of 2020. Well, that concludes today's ceremony. I want to especially thank Jim Allison for the insights he shared during his commencement address. And thank you to everyone for joining us remotely. Most of all, congratulations to our outstanding graduates. We look forward to the many contributions you will make to biomedical sciences in the years ahead. And to all of you, thank you for attending, and please stay safe.